Yeah, thank you for having me, Jackie. Thank you for the committee for having us share this data. This is actually an update from a presentation 2 years ago here, in which we presented the early part of the 2A study, the PK data, and the safety data from the dosing portion of the study. Before I go too far, I do want to acknowledge our CEO and founder, Dr. Dan Abrams, who we wouldn't be here if it wasn't for his, I think those that worked with him 10 years ago, to acknowledge his rather stubbornness to move this therapy forward. So since I'm up here presenting his, the result of his work, I want to acknowledge him. So first, just kind of talking about what the therapy is and what the platform is. So what we're doing is doing direct ICV delivery of VALPROATE for treatment of temporal lobe epilepsy. The idea of delivering directly to ICV is one, to avoid systemic exposure, decreasing the associated adverse events with that, allows a high drug concentration to be delivered directly to the brain structure of interest. And to do that requires proprietary drugs as well as proprietary devices. So from the drug perspective, to formulate this is actually a pretty big ask to be able to formulate the drug at the dose that we want, the pH that we want, et cetera. But then also to get that drug to play nice with all of the device components is a pretty big success story for our chemists in the company. From a device perspective, what we developed was basically a single catheter that would deliver a drug to the ICV space, and then also have a second lumen for CSF aspiration, which allows us to be able to evaluate that CSF from a study perspective, to evaluate PK. When you think about long-term patient care, to be able to look for infection, biomarkers, et cetera. Again, I mentioned we talked about the PK a couple years ago, so I'm not going to revisit that. Other than to remind everyone that what we saw in that is a 20x higher concentration of VALPROATE in the CSF versus oral administration. And then conversely, a 6 to 10x decrease in serum concentration of VALPROATE, again, compared to oral administration. So particularly when we look at serum, going from typically a target dose of 80 micrograms per mil to see 10 micrograms per mil. From an implant perspective, pretty straightforward. When we talk about it being minimally invasive, we're talking from a neurosurgical perspective and other surgical options for refractory epilepsy. So minimal compared to certainly resection, DBS, RNS, and I would argue VNS, although perhaps because that doesn't penetrate the cranium, one might argue that. But simple procedure, once they've done a couple, it's usually under 45 minutes open to close. And if ever needed, easily reversible to look at other options. And you can see there in the image what it basically is a common drug pump implant in the abdomen, catheter run up behind the ear, and then connected to the cranial catheter. Now, having shown this in the previous slide, just to be clear, for the 2A study that I'm going to be talking about, we did not use our formulated drug or our catheter. It was Epilum formulated in the pharmacy. And then for delivery of the drug, it was a shunt. And then for CSF aspiration, we used an Amaya. So the 2A study, it was a phase 2 blinded dose ranging study. We ended up implanting following 5 subjects, so fairly small study. After a 30-day seizure baseline, they were implanted, had a 28-day recovery, and then went on to that blinded phase of dose ranging. So again, talked about that previously. So I'm going to focus on the open label extension study in this discussion. So in the open label extension, they increased the dose up to 200 to 220 milligrams per day. They're seen once a month, primarily for drug refill, and then every 3 months for additional neuropsych, MRIs, et cetera. Now, the 4 weeks we are working on, we know from a long-term perspective and therapy adoption, particularly from a patient perspective, having them come in every 4 weeks for a refill is not really ideal. So 2 things that we're working on immediately is first half of next year, we'll come out with a new formulation that will push that refill period out to 60 days. And also what we're looking at, particularly in the US where it's common for pain pumps, is offering home refills of those pumps as well. Baseline demographics for this 2A study, again, severely refractory temporal lobe epilepsy. These patients came in with a median of 28 seizures per month, range of 17 to 235. So these are fairly severe patients. They all had tried at least 6 oral ASDs. The protocol required 3. They all were 6 and above, and all had to have trialed oral VALPROATE. Starting with efficacy, Looking at 12, 24, and 36 months, we see very sustained efficacy with median seizure reduction of 91, 86, and 91% respectively. The month 3 was a little bit lower, largely driven by the one non-responder. She's the subject that came in at 235 seizures per month. So very severe. She's typically seen about a 30 to 35% reduction at this point. Took a little bit longer for her to get to that. Again, responder rate 80%. Coincidentally, that one non-responder is the one that has the best BDI and COLI reductions. She's doing extremely well from that perspective. And a couple hypotheses around there, although we don't really have the data around it, is one, at 235 seizures, she undoubtedly was undercounting even that number of seizures. Still probably undercounting, but potentially to a lesser degree. And additionally, anecdotally, she reports in the diary where they're able to make comments that her seizures are significantly less severe when she does have them. So median BDI reduction has seen a 59% reduction at 3 months. COLI, 33% reduction, and we are going to do another analysis on more current quality of life measures. From a safety perspective, it's quite strong. So again, 211 total months of therapy for these patients, median being 42 months. Drug-related TEAEs were brief and transient. The most common being sensation of feeling cold, hair loss, insomnia. These all resolved within 2 to 3 weeks. No drug-related events were graded as severe. No TEAEs result in temporary or permanent drug discontinuation. Interestingly, a lot of what might have been thought of as CNS events, we didn't see any tremors in any of these patients. We didn't see weight gain. So as we get more patients, it'll be interesting to see how that plays out longer term. CSF pathology shows no histological changes. Serial MRI evaluations every month show no structural changes. Now, one caveat here is for the 2A study, we didn't have MRI protocol. So we don't have the sequences to do the degree or robustness of volumetric structural measurements that we'd like to. So this is under gross evaluation. For the 2B study, we do have a protocol for MRIs that will allow us to do much more robust structural evaluations. No revisions still to date on the delivery port. Again, this was with the shunt, not our catheter. And then we had one Ommaya infection that required removal. So the 2B study, which is ongoing now in Israel and Australia, 5 sites in Australia, 3 in Israel. The goal is 52 evaluable patients. We currently have 29 consented and 12 implanted. Primary endpoint of seizure reduction compared to placebo, and then the standard safety endpoints looking at both drug and device related events. Secondary endpoints, responder rate, COLI, SSQ, BDI, BI, PGI, and looking at reduction in seizure severity. So overview of what the design of this 2B looks like. So we do 6 weeks of diary baseline. Following that and meeting the criteria there, they get implanted, have a 28-day recovery of that implant, and then randomized to either placebo or active drug. They go through a 28-day dosing, 4 different dosing levels up to the target of 220 mg per day, an 84-day evaluation period. And then following that evaluation period, they do a blinded crossover period. So those that were randomized to placebo then go through that dosing to go up to that target dose of 220. Those that were randomized to drug, they basically go through a mock escalation to keep everybody blinded, and then rolled over to long-term follow-up, and then eventually to an open label extension protocol. So summary here, phase 1b/2a, median seizure reduction of 91%, very sustained efficacy now through 3 years, no DLTs or events leading to therapy withdrawal, 20x increase of CSF drug concentration with that decrease in serum concentration. The phase 2b, we plan to complete enrollment first half of next year. And also, because it's proof of concept that we are able to deliver drug at high concentration safely to a target, we are going to be looking at initiating a similar study for IgE in 2023. We're currently collaborating with Praxis on genetic epilepsies, and this opens the door to look at additional CNS diseases as well.